AIDS was recognized as a new disease in 1981, although cases have since been identified from as early as the 50s. The virus was isolated in 1983 and is now known as HIV-1, following the discovery of a second AIDS virus, HIV-2. Closely related species have been discovered in various apes and monkeys, and it is likely that all these immunodeficiency viruses evolved from an ancestral type several hundred years ago. It is not known when humans first became infected, but it was probably long before the earliest AIDS cases came to light. HIV is a small and fragile virus. Inside are 2 strands of RNA which contain the genetic information for the virus. This is unusual, since most organisms, including humans, store genetic information in DNA. The RNA and viral enzymes are packed in protein to form the core. Surrounding this is a protein matrix covered by a fatty envelope through which project protein spikes. These allow the virus to attach to receptor molecules present on the surface of certain human cells. When HIV has attached to a cell receptor, its envelope fuses with the cell membrane as matrix and core enter the cell. The matrix proteins are lost, leaving the core. Inside the core, the reverse transcriptase enzyme converts the RNA to DNA. When it reaches the nucleus, the virus DNA enters through a nuclear pore and inserts itself into the DNA of the cell. This irreversible joining of virus DNA and host cell DNA is called integration and leaves the cell permanently infected. In this integrated form, the virus can remain dormant for long periods, perhaps for years. If the cell becomes activated, then RNA copies of the virus are made which migrate out of the nucleus. Some of these RNAs stay intact and can form new virus genetic material. Other RNAs, which have been modified, instruct the cell to produce virus proteins. Near the cell surface, virus RNA and protein collect together and assemble into new virus particles. These bud from the cell surface, becoming cloaked in cell membrane. The membrane forms the fatty envelope for the virus. The virus particle matures internally and the life cycle is completed. The new virus particle is now free to infect other cells. HIV is very changeable. During replication of the virus, there is potential for mutation, and in an infected person, new strains of HIV constantly appear. Whenever a new variant arises, the immune system takes time to recognize and respond to it. The virus not only mutates to outrun the immune system, but also attacks it directly. The main target cells for HIV are helper T lymphocytes and macrophages. The helper T lymphocytes are crucial to immune functions since they stimulate other immune cells, including killer lymphocytes, which attack infected cells, and B lymphocytes, which produce antibodies. Macrophages are large, wandering cells which engulf foreign material and are also vital to the immune response. Nevertheless, the immune system does mount a response to the virus. B lymphocytes produce antibodies against components of the virus, and killer lymphocytes attack HIV-infected cells. These responses may slow the spread of the virus, but fail to clear it from the body, and infection remains for life. As the infection advances, the helper T cell population drops, and the immune system becomes progressively disabled. Eventually, sometimes after many years, the body is left almost totally defenseless against infection.